Amino acid are the organic molecules that join in specific number and sequence to make protein. About 170 types of amino acid occur in organisms. However, 20 types of amino acid participate in making most of the protein. An amino acid is an organic molecule made of a amino group NH2, a carbolic carboxyl carboxyl group COOH, a hydrogen group H, and a side group R, which are the attach a central carbon atom. H2N RCH is the structure model of atoms of these carboxyl hydrogen and the side group R, which are the attached to a center carbon. Amino acid journal structure. Different amino acid contain different side group. For example, in amino acid glycine, the side group is H, and amino acid alanine, the side group of CH3 methane N2 H2N HC is HCOOH, and H2 hydrogen group of carboxyl, and this is the carboxyl carbon hydrogen group, carbon hydrogen H2 and amino carboxyl group. This is the carboxyl group alanine. Methane is insecticide and amino 2 amino acid. Sugar carboxyl group is, for example, we get carbohydrates through from the stems or cornstarch and other. So this type of the group H2 and NH2 is in use in urea amino acid and in the charcoal of the fleet of the and acid, it is a acid of dirt and fleet of cows, generally grains. Stuff source and functions of protein. Good source of protein include meats, mutton, beef, chicken, fish, eggs, and milk, pulses, beans, and etc. Proteins perform various functions in our bodies, including protein are an important part of a cell membrane. Some protein, e.g., collagen and keratin, make almost whole structure of cartilage, hair, and nails. Enzymes are protein that catalyze or biochemical reaction occurring in organisms. Some protein are hormones. They regulate body process. Hemoglobin protein transport oxygen in the blood. Acting the myosin protein are the main components of muscle cells. They are responsible for muscular contractions. Fibrin is a blood clotting protein that makes blood clot to prevent the loss of blood after an injury. Some proteins called antibodies, part of our immune system, defend the body against harmful pathogens. Lipids are the organic compounds that are insoluble in water but are soluble in organic solvent, e.g., alcohol, ether, benzene. They are composed of glycerol and fatty acid. Glycerol is an alcohol having three carbon atoms. Each carbon has a hydroxyl group. Each, each carbon, one, two, three carbons with each has hydroxy, hydroxyl OH and hydrogen H. Glycerol. Fatty acid, glycerol is a oil. Fatty acid are known as in chemistry oils. Fatty acid are long hydrocarbon chains and with carboxyl group COOH at the end. There are two type of fatty acid. Saturated fatty acid have internal carbon atoms bonded with maximum number of hydrogen atoms. They do not have double bonds between carbon atoms. Saturated fatty acid are solid at room temperature. Unsaturated fatty acid have one of or more double bonds between carbon atom. They are liquid at room temperature. Here is double bond between oxygen and the hydrogen, and there is carbon carbon double bond, and the oxygen and carbon or all other have the single bonding. Main structure: saturated fatty acid, hydrocarbon, and three atom of hydrogen and one of carbon with carbon carbon bonding and a hydrogen carbon bonding. Cholesterol, the fatty acid, carbon hydrocarbon bonding, fats and oil general structure. Cholesterol HCH, carbon hydrogen bond, carbon hydrogen is a chain of phospholipids generally journal structure. Phospholipid phosphate group phospholy phospho, phospholipids journal. What main group of lipids? Fats and oil are the most familiar lipids. They contain one cholesterol and three fatty acids. Fat contains saturated fatty acid, and the soluble are the saturated, and the unsaturated is that they do not dissolve or soluble in alcohol, and so are solid at room temperature, e.g. animals fats of the. On the other hand, oils contain unsaturated fatty acid and so are liquid at room temperature, e.g. example Greta plant oil such as olive oil, corn oil, and coconut oil. Phoso, pho, phospholipids, these lipids make the core of the all membranes. Phospholipids molecules contains often cholesterol, 2 fatty acid, and the phosphate group. Source and functions of lipid source: more the cholesterol, These they causes more than the actual value causes the cholesterol test. Source and function of lipid source of lipids from animal are meat and dairy product, while the sources of the lipids from the plants and nuts, seeds, oil, olive oil, etc. Plant synthesis oils and store them in seeds such as sunflower oils, coconut oil, groundnut oil, and the corn oil. Functions: lipid are the most energy rich biomolecules. They serve as a long-term energy reserve in the form of fats, as in adipose tissue. When the body requires energy, these stores stored lipids are broken down to release fatty acid and cholesterol, which can be used as fuel for energy. Lipids are essential component of a cell membrane. Lipid acts as insulator and protects vital organs from example adipose tissue surrounding organs, provide cushioning and heat insulation. Some lipids help in the synthesis of the hormones. Steroid hormones are derived from a lipid, i.e., that is cholesterol. Lipids help in the absorption of fat, soluble vitamins A, D, E, and K, and the digestive system. Nucleic acid are the biomolecule that are the composed of units called nucleotides. All nucleotides is the nucleoids is the made up of T cell and is made up of three component pentose sugar ribose or deoxy ribose nitrogen space phosphate group PO4. Phosphate was a pond powder ash and the pentose and base structure 015321. Phosphate group nucleoid journal structure nucleoids and there are two type of nucleic acid deoxyribonucleic acid DNA is made of nucleoid deoxyribonucleides is in this nucleoids the pentose sugar is deoxyribose while the nitrogen base may be adenine, a thymine, C, cytosine, C, or guanine, G. In 1953, US biologist James Watson and British biologist Francis Crick proposed the double helix model of DNA. According to this model, DNA is a double helix molecule. It is made of two strained nucleoids. Both strained are collide around each other. The nitrogens based of one strand make hydrogen bonds with the nitrogens bases of the opposite strand. Strand हमारी this is the strain one strain two, and these are the N base cytosine. Adenine is by C, A, and G guanine, and the thiamine figure double helix model of the DNA. DNA is the pairing of nitrogenous bases in specific that is adenine of one strain forms a pair with thymine of opposing strain. Similarly, cytosine forms a pair with guanine. There are two hydrogen bond between the adenine and the thymine, and the three hydrogen bond between the cytosine and guanine. Function of DNA. Function. DNA contains the hereditary information. This information is in the form of sequence of nucleoids. This sequence determines in the order of amino acids. During protein synthesis, the segment of DNA is in which the dead protein in waste proteins is called DNA, in which the but presence in human body, which is the sequence of the nucleoids determine the synthesis of a protein polypeptide chain. It is called a chain. During reproduction, DNA is passed from the generation to the next in this way. DNA carries the heredity information to the next generation. Ribonucleic 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 acid RNA is a single strand. It is strand consists of ribonucleotides. Ribonucleoids contain ribose sugar instead of deoxyribose in a new ribonucleoids. The nitrogen base may be adenines, uracil, cytosine, or guanine. There are the three type of RNA message messengers. mRNA carries the genetic information from DNA to the ribosomes during protein synthesis. Transfer the RNA and transfer specific amino acid to the ribosomes and ensuring the correct sequence during protein synthesis. C. Ribosomal RNA smaller RNA consists constitutes the structural and the functional components ribosomes, the cellular machinery of for protein synthesis. There is a no living organism or a part of protein. It is, for example, it cannot move nucleoid or strain, but it found in a living organism like human. The working of DNA and the RNA, the DNA molecule is a chromosomes consist of thousands of non-nucleoids along the length of DNA molecules. There are specific segment called chains. Each chains consists of a specific sequence of nucleoids that carries information for the synthesis of specific protein. During the working of a chain, the specific sequence of the DNA nucleoids is copied, the copies in the form of molecule of messenger RNA, small m RNA. The process of the making small mRNA copy of DNA is called transcription. The mRNA carries the sequence of its nucleoids after ribosomes. The ribosome reads this sequence and joins specific amino acid to form a protein. This step is known as translation transcription segment of a DNA. Chromosome ribosome translation mRNA moves to cytoplasm. Division of working of DNA, also called the central dogma.